Thank you very much. Yes. Right. Very nice to be here, even virtually, kind of at the High School of Economics. Maybe first a few housekeeping notes. So we will hold it in English, but feel free to ask your questions in any language, really. If there are some kind of conceptual questions, maybe best to hold them to the end. I know we will have around 20 minutes for questions in the end. But if there is something that you don't understand while I explain things, maybe vaguely, feel free to interrupt me either just by muting yourself or via chat. I hope I can see it. Also, if some of you feel comfortable to switch your video on, that would be actually quite nice just to see maybe a few faces and kind of talk to someone, not just to my laptop. So please feel free to do it if you feel comfortable. Oh, hello. Thanks. Cool. So, all right, let me get started. So my name is Anna. Looking at the participants chat, I know that I know some of you, and then I guess some of you know me. So I've been studying in Moscow State University. I've been working on natural language processing research mostly with Konstantin Vorontsov. And then I transitioned to High School of Economics to do my PhD, also staying with Konstantin Vycheslav, which is my science advisor. And then after doing the PhD, I joined DeepMind about 3 years ago now. So I've started with some language projects at DeepMind, and then I thought it's time to transition and try something new. And I tried to join the bioinformatics team, which is part of the DeepMind bigger science team. And I'm very glad I did, because I think that was kind of my kind of long-term goal and kind of dream to work on something that I feel is really important, like for the science and for humanity, and try to apply my knowledge in that area, like especially given that I was not trained with any kind of natural sciences, so I don't have any biological background. So it was much fun to try to apply computer science background in a new area. We have a very disciplinary team, as you can see from the number of authors on just this slide. We have maybe about like 20 people working actively on this paper and the method for this paper. Some of them have, more biological and some more computer science background. Right. Let's see. So I am going to present the method, the AlphaFold, which was the method to solve protein structure prediction, which was very successful at the CASP competition for protein structure prediction recently. And subsequently, we published the Nature paper and released the open source code for biologists to use. That's what my presentation will be about. Cool. So I'm not totally sure what's the background of the audience, but I bet we definitely have folks as myself from computer science background. So I'm kind of thinking of myself 3 years ago. I didn't know much about proteins, other than like they are important and that they exist. So a little bit intro to that. So proteins consist of chains of amino acids. Amino acids, there are a lot of types of those, but mostly 20 types of amino acids are most popular. So you can think about the chain like a sequence in the alphabet of 20 amino acids. Usually you have sometimes you have multiple chains in a protein. They might form multimers. But now a chain will fall to a 3D structure, and 3D structure is very important for understanding protein function. And that way, solving protein structures has been an essential problem in biology for over 50 years. It's not only important, it's also very hard actually to solve by existing methods. So it's reasonable to say that it's worth a PhD kind of timeline to solve even just one protein structure. So a PhD student might work on all those steps on this slide to finally come up with a 3D coordinates of atoms in the protein based on the amino acid sequence. And our method is approaching the same problem, but obviously with an AI-driven approach. So why does DeepMind work on this at all? Like why DeepMind to work on this problem? Well, DeepMind is pretty serious about its mission, and it wasn't that kind of apparent to me before I joined, but that's how it feels internally. DeepMind mission is to solve intelligence and advance science and benefit humanity. So, like from these three rather kind of loud statements, you can see that the protein structure task fits really well in the scope. So that's why we work on it. So now this summer, there was one of regular HASP competition, which is a assessment of protein structure prediction. And in this competition, AlphaFold showed really high performance. So on the right, you can see the GDT, which might not tell you much, but this is a standard accuracy metric for predicting, for assessing quality of structure of a protein. Roughly, it says, so when you have two proteins, you first have to align them in some sense in 3D space to measure how close they can be aligned. And then GDT will roughly say what is the percentage of residues or like the percentage of amino acids that can be aligned within certain small distance measured in angstroms. So importantly, GDT of 90 is considered by CASP organizers compatible and comparable with experiment quality, the one which would take maybe years to finalize. So that's a big deal. And on the left, you can see a couple of just examples of structures from this competition. The left one is maybe one of our favorite ones, since it was one of the largest, and of course took a bit of care in the competition. And afterwards, we like to test kind of our limits and our scale on it. And you can see that even for this big target, for this big RNA polymerase, the green and the blue are aligned actually quite well. Cool. So actually one more important point just to say on this slide, we are honestly very grateful to CASP organizers and for the community for running this competition and for doing this over 25 years, I think. And without the community, it wouldn't be able kind of to define the problem that clearly and also to assess the quality of competitors. So that's absolutely great and tremendous effort. And that's great that in this area it happened and it exists. Cool. So now just to briefly what is going to come next, I'm going to talk about AlphaFold method, like main intuition and main structure bits of the architecture. Then I'm going to talk maybe about my favorite part, about how to interpret AlphaFold predictions and how to try to interpret the network itself, like what is doing and why. And then finally, I will talk a bit about where to try things if you're interested. And and Q&A. So maybe now is a good question just to interrupt me if you have any immediate questions after intro or anything at all. All right. If not, feel free to post on chat. I now see it. And I will move on. So how AlphaFold works. All right, before how AlphaFold works, what is the inputs and the outputs for AlphaFold? So we briefly talked that the main input is amino acid sequence. I will interchangeably say amino acids or residues, meaning kind of the same thing. In this talk, and on the right, you can see like an example chain. Then another important input for the method is derived from this chain by searching similar sequences in a big protein databases. So on the right, you can see so-called multiple sequence alignment, MSA. And this is evolutionary related sequences. So they might have some mutations and thus the sequences have diverged. However, you might expect that they still fold into similar structures. So it might be useful to provide those sequences to the network. And actually, that's what many other networks and methods do as well. So there is nothing too surprising about that. And then the data that the model uses for training is mostly labeled data of the Protein Data Bank, PDB. And also, and we will talk a bit about it, we use unlabeled data in kind of self-distillation approach. And after that, the output is the 3D positions of every atom in the protein. So on the right, you can see kind of an example of a folded protein chain and how it might look like. Right. So speaking of this MSA, or this multiple sequence alignment, just digging a little bit more into intuition, why this might be important. So the input sequence, we have some research sequences in genetic databases to find multiple sequences. And then the coevolution says us that residues that are in contact in the folded structure must mutate together. So why so? Well, because if single residues mutate independently, then they are likely to break the contact. And it means that we'll break the particular local structure and possibly the function of the protein. So the mutated organism won't be able to survive during the evolution. So assuming that close residues that are in contact mutate jointly, we can see that there is some important information to extract from columns in the MSA and kind of implicitly use this data to learn about contacts in the structure. So that's kind of a high-level motivation why evolution data might be important as an input. Now, a few kind of high-level words about how the network works and what we've been trying to put into it when designing this. So first of all, it's end-to-end system producing a structure from the amino acid sequence. And this is opposed to an earlier AlphaFold 1 solution that we had a couple of years ago, and also opposed to many other systems in this field where interresidue distances are predicted, but not the final structure itself. Then importantly, we've been trying hard to put physical and geometric insights into the network architecture itself. So kind of speaking of inductive biases in machine learning, it might be important to see what architecture will fit best to the problem. And in this domain, so the previous system, AlphaFold 1, was actually kind of a mix of off-the-shelf methods with convolutional neural network kind of taken from computer vision and put into the middle of the network. But then here we've been redesigning everything from scratch, and the model is based on attention technique, which originated in language, but still language, pure language transformers were not working well for this task. So it turned out to be important to put kind of geometrical and physical knowledge about proteins that we might have. And as a concrete example, it's important to de-emphasize the sequence order of the residues, because actually it's not that important that this residue and that residue are far away in the sequence, let's say, if they turned out to fold and be close in the space. So if they turned out to be close in the 3D space, it's super important to model them together and kind of let them communicate with each other and update their 3D coordinates jointly. And therefore, it's not enough just to reason about sequences during the inference, but we need some sort of residue-to-residue communication, like all residues to all residues should be able to talk and to build this graph while iteratively refining this graph. And we will get to concrete like implementation of this in a bit. Okay, I'm seeing some question on chat about Msa parameters. So maybe it's a good point to answer this. So as I said, the network is trained end to end. So we will see the architecture and like the concrete embeddings are just parameters of the network, which are trained via back-to-back gradient, gradient-based approach. So that's kind of up to the network to decide which MSA sequences are important and which are not. And we've been noticing that it's actually not harming to add extra MSA sequences as the input, and the model will be able just to throw away some noise and be robust to it in some sense. So speaking of the inputs to the network, those are some standard databases, and we use some standard search tools over those databases. And then the network itself comes. So let's build it with you, like step to step, and have a full complete picture. So first step, input sequence, search of those MSA, multiple sequence alignment MSA. So here it's like 2D array of sequences by residues. By sequences here, we also depict some nice organisms. We spent a good deal of time drawing those nice little animals. So highlight for those. And then the second important input, as I've been just talking about, is pair residues. So residue by residue matrix, which is initialized somehow simply, but then importantly, the network has this capacity to update those residue pairs and reason about them. And finally, it is also possible to have as an input templates. So templates are structurally similar proteins with known structures. So we can just, again, extract them, search them, and find them with some regular open source tools. And we can use those. But it turns out in our ablation study that templates are actually not tremendously important. And if you just throw them away, you just get like one GDT drop at most. Right, so once we have inputs, the main network comes, which we call evoformer. It is 48 block network based on attention. So basically in every block, we do a lot of sequence wise attention and also residue wise attention. And we will describe it a bit more in a bit. And importantly, after this evoformer blocks, we expect those MSA representations and pair representations to be highly processed and contain some useful information about residue locations. And like already those outputs of this evoformer should be enough to predict the final structure. So after we have that, we have just really shallow structure module, which has 8 blocks and very simple shallow architecture, which just takes those highly processed embeddings as an input and turns them into concrete coordinates of a structure on the right. And it, like as a kind of extra trick that turned out to be super useful, it turned out that if you take the outputs of the network, the 3D structure, and put it back as inputs to the network, then the network gets kind of another opportunity to reason about the structure and refine its own predictions by the same architecture, really. So the weights are shared between all those recycling iterations. So basically, it's also kind of a cheap way to make the network deeper by running the network 3 times, every time passing its own outputs from the previous recycling iteration as inputs. And we found that that's kind of boosts the accuracy quite a bit. Right, I see there is some other question in the chat. The residue pairs matrix isn't inferred from MSA. From the scheme, it seems it derives directly from the input sequence. So I guess that's the question. Okay, so the residue pair matrix on the left here in my inputs is just it's just like kind of 2D outer sum of the sequence or something like that. But then importantly, I hope I actually will have it. Importantly, during the main evoformer blocks, MSA representations and pair representations talk to each other. So there are updates that influence both ways. So actually, yes, MSA representations will influence residue pairs matrix during those 48 blocks.
And what is at the beginning? What is inside this residue pairs matrix?
I think it's just outer sum of like one hot encoded amino acids or something as simple as that. So basically just some way to initialize. So basically all information there is like the identity of amino acids. So that's basically like kind of a placeholder to have these parameters, to have this space to update them later. So pair representations with the blue ones on the right are actually the interesting ones.
And am I correct that during this multiple sequence alignment, you search in the database, you search some sequences that are similar to your sequence in some places, right?
Yes, so we actually use kind of third-party tools which do like various heuristics, how exactly to search. So it can turn out that we will have kind of partial match, which is similar just for one domain, let's say, and not similar anywhere else.
And I don't understand how would you add at the recycling, how would you add outputs to the inputs? So how this is done?
Yeah, that's a great question. So we have a 3D structure. So we have 3D coordinates of all atoms. Indeed, how do we turn this kind of array of numbers, 3 by number of residues, 3 by number of atoms, how do we turn it into something which we can add to the inputs as plotted on the slide? So we, for the residue pairs case, we compute what we call distogram. So just distances between the residue pairs according to the structure. Because given the exact atoms, you can say, okay, the Euclidean distance between residue A and residue B is this. And this distance matrix of residue pairs is added to the inputs. And I think we, for the case of MSA representations, we simply add the final MSA representations from before the structure module to the input ones. So that there it's even easier. So we've experimented with a bunch of different ways, like whether it's enough to add just the structure or whether it's also useful to recycle embeddings. And I think we recycle all of them at the moment. Seems to be helping a bit.
Thank you.
Thank you. Do you realign MSAs in the course of the refinement process? Yes and no. No, in a sense that we do not search for new MSAs and we do not realign them. But yes, in a sense that it turns out that in the outputs of those search tools, we have actually too many MSA sequences that we can't process at once. So we have a bunch of kind of heuristics how we sample MSA sequences and how we cluster them such that the network sees the most representative clusters and also some additional not cluster sequences. So it turns out to be useful to resample these bits on every recycling iteration. So yes, we resample there. Cool. And when we don't have enough of them, I guess we just, sample every time the same ones will be sampled, and it's fine. And when we have absolutely not enough of them, like maybe less than 10 or less than 30, we do see that the method can get worse on those really shallow MSA cases. Cool. So apart from predicting the structure, it turned out to be very useful to predict some sort of confidence metrics by the model itself. And in real kind of biological applications, it's very valuable to know where to trust AlphaFold and where not to trust AlphaFold. So we predict two kind of measures for that. One is per residue and one is pairwise. And I think I will not talk more about it now because we will have a whole section on it in a bit. But the important point here is just that it's coming from the same architecture with the same weights, just final heads are trained to predict confidence and really shallow ones. Cool. So instead, let's dive into maybe the main block, which is evoformer. So the inputs are the MSA and pair representations. Then we have attention. Actually, we have what we call grid attention. So in the case of MSA, it will be attention for each row and then attention for each sequence. And then we have outer product, which provides a way to inform the residue pair representations by MSA representations. So that should hopefully answer the previous question on whether those two kind of towers of embeddings are in sync. So they are in sync, and there is actually kind of a line. It should be like another error, the opposite direction, where we bias the MSA embeddings by pairwise embeddings. And then maybe the most interesting bit is what we call triangular attention on the pair stack, on the pair embeddings. So to have a kind of idea what this triangular attention does, let's think about our residues as about nodes in a graph. So edges are pairs of residues then, and we know that we want to infer this graph of distances between residues, while at the same time we don't know the graph. So we have to kind of jointly do that. And the idea here is simply that there are a lot of constraints in this graph. For instance, if we want to update this green edge, it makes sense to look at the red edge and the yellow one and say, well, the update for the green edge, let's call it IK, should depend on the updates on the distances on the two other ones. So basically, this is like to ensure consistency. You can think about it as a triangle inequality or something roughly similar, basically saying that we cannot just pull all those residues in any directions we want. We should at least provide a way for the network to stay consistent and try to make those cycles of three residues work reasonably. So each update there is just attention coming from from transformer, but honestly even from earlier work before transformer. So in the attention we will update, we will learn a few matrices of parameters, and we will update the embeddings in these representations. Well, embeddings in this in this matrix. Then after the evoformer block, we have the shallow structure block as I promised, and that one is a fun one in a one particular sense. So it turns out it is rather inconvenient to parameterize the protein chain, protein backbone in 3D space, and like, okay, maybe I should just say first a few words about the picture, so you can actually see protein backbone on the left, consisting of certain atoms. You might expect some side chains to be also present there. You can see like three angles that determine the geometry of the protein backbone, and there are multiple parameterizations on how to encode this protein backbone. So you can think about bond lengths, and you can think about angles for each next bond, right? But interestingly, it turns out that it's useful just not to tell the network at all about the fact that this is going to be a protein chain and that it has to have some reasonable bond length. But instead, we treat this just as a bag of atoms, really, well, bag of residues. And we let the network to infer the rest. So yeah, we think about this 3D rigid bodies, as you can see those triangles on the picture, and then the network is allowed just to combine those into any protein backbone, and it turns out that it learns reasonable bond lengths. Importantly, it's end-to-end folding, so the gradients are propagated through the whole architecture, and it's 3D equivariant meaning that any transformation and rotation will not change the result. So it's equivariant architecture. And then finally, the end of the structure model is the FAPE loss. We call it FAPE, frame aligned point error. This is if you think about aligning the structure to ground truth, you can use each of those rigid bodies to align your structure to ground truth. And for each of those, you can just compute Euclidean distance between atoms and ground truth for all other rigid bodies and come up with the loss. And like you sum it over multiple alignments. Again, it turned out that all kind of more normal losses usual in machine learning were working less good. So that one was kind of customized for protein domain. And then finally, we actually, maybe on some of these slides, I should show that apart from those protein backbone, we also predict side chains by predicting angles that will just enforce their position. And with that, we will have coordinates for all atoms. But we might end up with some minor structure violations, meaning that, for instance, some atoms might end up on top of each other, right? On the right picture, you can see that the steric violation is something which is not physically plausible and cannot happen in nature just because like some atoms are sitting on top of each other. So finally, before we provide the final result, we run the network, we run the output through relaxation procedure where we use, again, some kind of third-party standard tool to slightly shift the final positions and try to avoid those kind of steric violations. All right, so that was kind of very quick and brief run through all essential components of the network. But then more, yes, there are, and which of them actually matter. And we've spent, honestly, like quite a fair amount of time this summer, and me personally, running various ablations, and I, to make this plot on the right, where we try to compare, okay, now with the final network, can we switch off these bits? Can we change that parts? And which of them matter the most? So the very brief conclusion seems to be that there is no single improvement, which is clearly like the one breakthrough, which has to be the key for the accuracy gain. It's more like a combination of several things, and more importantly, they matter together. So at the bottom, you can see an ablation where we knocked out multiple bits of the network, and then it finally got worse in terms of the GDT and some other accuracy metrics. So I'm not going to dive into those ablations in details. They are in our paper if you're interested, but I just wanted to highlight one particular, just because it's kind of a nice bit of the training procedure. So actually in these ablations, we compare with baseline, but our real network is baseline plus self-distillation training. So self-distillation training is a way for us to use unlabeled data. And there is, of course, much more unlabeled sequences than labeled sequences with provided structure. So we first train our network with the same architecture that I have described. We train our network and then we use it to predict structures for a huge data set of sequences. And then after that, we use those predicted structures as training examples, as additional training examples in our second training of the model. And we sample basically from the real training set and from this created set of our own predictions with some weights. And we do see a noticeable boost on any metrics with that approach. Okay, I see some questions on the chat. Are side chains predicted only once or at every iteration? So side chains will, which iteration do you mean specifically, since we have a bunch of various iterations? So if you mean recycling iterations, they will be predicted at every one of them. But other than that, if you don't think about recycling, then side chains are really just a small, shallow network in the end there around structure module, which again takes in highly processed embeddings and outputs the torsion angles. And then there is kind of hard-coded bits how you produce concrete atoms from atom coordinates from all the torsion angles for the side chains. Mm-hmm. Yeah, for recycling, it's you can think about it as like everything is repeated and please the network try again. All right. I have last slide in this section of the architecture itself. So I just want to refer you to our supplementary materials, which has 60 pages of those algorithms and diagrams that we've been drawing over the summer. So yeah, feel free to consult for any details. And also the open source code is another kind of opportunity to see what exactly is happening where, since it's like the best way to reproduce things. All right. Are there any questions on this section about general architecture before we move on?
Yes, I do have a question. Can you please go back to the slide when you are talking about gradient descent? You used some other technique instead of gradient descent. I think it was next slide, or yes. What does it mean? It it was previous slide. Yes, end-to-end folding is another way, another technique to feed the model, or what do you mean by that?
Sorry, yeah, I think yes. Basically, the context for this slide is that our previous AlphaFold 1 model was a model which outputs pair residue distances, and then after that you have kind of gradient descent based search to find the final structure. But here we don't do that, and we do like what you might think of like as you train any other neural network, where you just have a loss and you have back propagation from your loss for the parameters, and then you have update step and you update your parameters and you go on. So yeah, so this is obviously still gradient-based approach, but the point is simply just that it's end-to-end, and you just update your parameters as like any large neural network in any other field. So like no extra search module.
Okay, thank you.
What do you mean here that it is a 3D equivalent transformer? What is equivalent here?
Mm-hmm. It means that since we are talking about 3D objects, we would like to be invariant to rotations and translations. So what is meant here is that the architecture itself is robust to these things, and we don't need to kind of that we do account for the symmetry of those things.
And you just impose a coordinate system, like you predict some angles, and this is why it is invariant, or something else is used.
Yeah, I think, and also like all the updates which are used are also equivalent, right? So like we do not break this property while computing next updates within the structure module from the previous ones.
Thank you.
Yeah, actually, this one, like the structure model, is also kind of there is like an algorithm in the supplementary materials on that. So I think it might be nice just kind of to see it line by line, or maybe that's just because I'm so glad we finally wrote it down. That's why I'm advertising it that heavily. Can you explain again what is the output from the structure module? That's simply all the atom coordinates. So as we've talked, a protein consists of these residues, which come from different amino acids. And then before we talk about like sequence of these residues, but in the output, we are interested in atom coordinates for all atoms in the protein. So the output will be simply a matrix of coordinates 3, x, y, z by number of atoms in the protein, including all side chain atoms. All right. Maybe I will be slowly moving on to the next section of what have we built and how that might work. And please feel free to continue post on chat for any other questions. All right, so I will start with kind of 2 estimates of confidence that will help to use AlphaFold predictions. And then I will talk about how we tried to kind of probe the network a little bit to understand how exactly it works. So the first one, predicted Lddt. It's quite a funny name, which makes many people confused. So let's first figure out what is Lddt. So its proper name is Lddt alpha, also with first L not capitalized, because someone who did it first time many years ago did that, and now we have to comply. So Lddt alpha is a score about local correctness of the structures. So this is a measure of local, not confidence yet, since we are talking about Lddt itself. It's the percentage of correctly predicted interatomic distances. If you compare, if you compare your predicted structure to the ground truth structure, it's simply about how many paired distances you have got right. So there is nothing about alignment of the structures and like superimposing them as other metrics do. And that means that you just care about, okay, for this distance between these 2 atoms, have I got it about right? And it means that it measures local correctness. And with AlphaFold, we predict this measure for every residue. So it turns into local confidence measure of our predictions. And here we can see a concrete example. So the structures are colored by the predicted Lddt. We note that with pLddt higher than 70, this is a good confident prediction. For instance, it might be some good confident domain. With pLddt less than 50, that's usually not a structure prediction, but a disorder prediction. So basically, these residues are likely to be disordered in nature as well. And it turned out to be a good kind of indication for biologists of the disorder. And if pLddt is particularly high, like higher than 90, it starts to be reasonable to investigate side chains or things like active site details. So basically, that's kind of indication of really high quality structures there. And it has been quite fun to observe how people start to use those things on Twitter and post their predictions. So one of my favorite Twitter posts is, I think, don't trust the spaghetti, trust the fusilli. So you can see that you should not trust these regions of disorder, but alpha helices are fine. So the blue bits are pretty good here. Now, there are some issues with this metric coming from the fact that it's a local confidence metric. So if AlphaFold is confident about each particular domain, it doesn't yet mean that interdomain relations got right. So to predict confidence about interdomain positions, we've worked on so-called predicted aligned error estimation. And let's maybe look at this example right away. So imagine you align your prediction at residue Y to the true structure, and then you measure the error that you get at residue X. So it's going to be pairwise metric. On the one axis, you have kind of the residue for which you align. And on the other one, you have residue for which you measure the error, just in 3D. And here we color by these errors. So for this example, we have two confident domains with dark green, but actually interdomain correlations are not that confident. So like if you are going to explore the structure, probably you shouldn't be trusting too much. And indeed, in the picture on the left, you can see that there is some flexible linker in between. Now, turning into something about how actually internally the network works, we've been very curious what's going on there in the intermediate stages. So we've just talked, we have 3 big recycling iterations, and we have 48 evoformer blocks. So if we multiply one by another, it's like 150 roughly layers of something. What are the embeddings there? Can they actually predict this final structure maybe over there as well? So what we did, we just freeze the parameters of the whole network, and we train auxiliary probing heads, small structure modules, and we apply them at different intermediate layers. And on the left, we see just examples how accuracy increases over the layers. For some particular simple proteins, it will just jump high right away. For some other, and this is the green one, is orphate protein from SARS-CoV-2 protein. So this one increases very slowly, and actually for that one, it would benefit to have even deeper stack of layers. And I think I don't have this slide in this PDF just because, it was tricky to import a GIF, but you can find it at supplementary information for our paper. It was quite fun to see how the structure actually emerges. So you can imagine you start with like everything together in one place, and then the residues slowly get pushed out and you come up with the full structure. For some proteins, we also see a hint that they are searching over multiple conformation and then pick one. Okay, and then maybe final slide in this section. So we fold protein chains. So everything we've been discussing is when we take amino acid chain as an input, and then we get something as an output. But in nature, we often have much more complex structures out there. So for instance, we have DNA binding. So we can have a protein and DNA which binds to a protein, or we have a protein plus a ligand. So a ligand can be some small molecule that, like heme, for instance, that binds to a protein, and together they form some complex. And it can be that, and it is a fact that the protein can take multiple conformations depending on whether it's binding to a ligand, let's say, or depending on temperature and many other conditions. So all these things are actually not provided as an input for the AlphaFold system. And it's interesting that the network is actually able to implicitly model this missing context. And for instance, the picture on the left here is is an active site where zinc would be binded, but it wasn't provided to the network. But still, you can see this kind of 3 arms correctly inferred in 3D space. And in nature, though, that's because you are going to hold a zinc there in the middle. And similarly, on the it's a trimer structure. So there are three chains, blue, green, and red one, and they fold all together. And here it was possible to fold them correctly just by one chain. But kind of one of the future work directions is to really extend to all those conditions and adjust for them. All right, I see some questions in the chat, so let me pick them up. What was the process of architecture design refinement?
We could, in principle, also postpone this question till the end of the talk, if you prefer.
Oh, I think I don't mind. Right now, I think I have not too many slides left, so I think we will be on time. Yeah, and if the author of the question wants to jump in and ask it while I'm reading it, it's also fine. Otherwise, just reading, and I think everyone can see it in the chat, right? Yep. Yeah, I meant just like to unmute yourself and ask the question. But I think I am now up to date. Could finally read it. So yes. So what was the process of architecture design refinement, especially given that we have so many ideas mixed together, and some of them are intuitive and some of them are less intuitive? Yeah. So that's a kind of a good and conceptual question indeed. So one thing to say. We had kind of several eras of architecture where someone was just coming up with next good idea, which really worked well. For instance, as those like triangular attention, and then that's how like those kind of major bits came in. For many other smaller ideas that I mixed together, it was really driven by a kind of culture of a leaderboard in our team. So we have been working as a team of many people, and we always had state-of-the-art architecture currently checked in at head in our like internal repository, and we've been pretty careful to like when someone else comes up even with a small idea to put it on top of the other changes and incrementally grow our GDT. I think I don't have a plot at hand, but it's kind of rewarding to see how GDT was incrementally growing, sometimes with breakthroughs, sometimes with like incremental improvements. And that's how that evolved. So yeah, so a mix of kind of small and big changes in that way. And then for Hipaa parameter tuning, let's say, I'd say we were not super careful about that. So for many ideas, we just tried a shallow sweep of several things. Some numbers worked well and we settled on them. And indeed, when towards, like, let's say in ablation studies, we tried to be a bit more cautious about it. Usually it wouldn't give that much gain. So we thought it was fine just kind of to shallow search the surface and settle on something. Thank you for the good question. All right, let me then just walk through the last section and then we will have enough questions time, I think. Cool, so now we somewhat know how AlphaFold works and what to make out of it predictions, where to take them. So first we had a big release summer. First, we released AlphaFold protein structure database. So that's a collaboration with our amazing collaborators at Yambo EBI, where we host a website with AlphaFold protein structures, where everyone can simply search for their favorite protein and see outputs of something like what you see here on the screen. So they contain predictions for 21 model organisms. It's a human plus 20 other organisms important for biological research, and have plans to expand it soon to bigger data sets and eventually cover all of UniRef90, which contains around 100 million structures. So basically like the whole proteome, like all proteins for which the structures for which the sequences are known, roughly. So that's exciting. That's coming up soon. Next, if there are some proteins particular interest, there is open source code, and it is a bit of installation process. Obviously, everything is within a Docker container, but you would also need to download genetics and template databases, which can be quite heavy. So there are setup instructions, and we've noticed that, that like it took some time for people to download those things. But I think now most of protein research laboratories just have it on their clusters, download it, and we see increased statistics of using our tool just through open source code. And finally, if you're not that motivated to run it on a cluster or anything, you can just simply try AlphaFold Collab, which is, yes, a simple way to run something on a machine in the cloud. So you don't need to download anything. You just enter the sequence to fold and can play with some predictions that it produces. And just to close here, it was really fun to see how different biologists react, mostly on Twitter, sometimes saying that, like they were really curious to see how that structure looks like, and they could get it from AlphaFold before getting from an experiment. So that was definitely fun. And that, of course, wouldn't be possible without really interdisciplinary team coming together and working really in this close collaboration, as I said, like on one code base and one leaderboard and so on for a while. This is a thank you slide for everyone involved in this research. And now I am officially open for questions.
Thank you, Anna, very much. There is already one question regarding the ablations in the chat. And meanwhile, you could write other questions or maybe raise your hand, and then I can unmute you, or you can unmute yourself and ask the question.
Mm-hmm. Sounds like a good plan. Thank you. For ablations, the models were not retrained with ablations, just inference was performed. Is it correct? No, I'm glad to say this is not correct. So that was my fun summer to retrain all those models from scratch. They were retrained without distillation, though, as I have noted, because distillation because of course, like to do it properly with distillation, it would mean 2-round training for every model, which would be too computationally costly. But other than that, the models have been retrained, and also we had 3 random seeds for every model, just because we do see some variants. Sorry, just trying to find the plot. So 3 seeds for every ablation, and there should be some more in the supplements.
Okay, there is another question. The first one was Nazar. So Nazar, feel free to unmute yourself.
Okay, cool. I have multiple questions, honestly. Thank you for your presentation. It was very interesting. For my first question about matrix P-L-D-D-T, did you feed it somehow for some loss, or some additional information that you can take from the model during the evaluation?
Good question. So yes, so it is yes to the first part. Yes, we have a shallow head that predicts LDDT. So it's coming all from the same stack of layers, really, like here. And you can see that we take the first embedding of the Msa representation, because that's the embedding that contains the target sequence that we are trying to fold. So we are taking this embedding and we are passing it to a super shallow, like MLP, if I'm not mistaken. And the loss is basically we are predicting a binned ground truth LDDT on this example. Since we do know our prediction during the training and we know the ground truth, so we can compute real LDDT, and then we can try to predict that score with this head. And we notice that it's actually very easy for the network to learn that, which is a little bit surprising. Like the network knows that it's wrong in some areas, and it's uncertain in these areas, so it cannot improve them, but it can predict that, oh, please don't trust this region. Did I answer?
Yes, you did. Thank you. My second question is continue this first question. We know that some proteins have wobbling structure. How did you? I know that in first AlphaFold you predicted some. You could predict multiple conformation of one protein, and how this metric Lddt is working on this wobbling part of protein. And could you use AlphaFold second for multiple structure prediction?
Sorry, did you mean like those bits that can have multiple conformations, or did you mean like flexible regions?
Multiple conformations.
Yeah, sounds good. Yeah, I think So for that, actually, I think I would. We have kind of a limited success for that to use multiple predictions. So basically, in our final system, we run the model 5 times. We run 5 slightly different models, and we produce 5 different predictions. And sometimes we do get diversity out of those predictions. So you can expect that those wobbling regions will fold, let's say, in this state in one of the predictions and in that state in the other of the predictions. But that's not always the case. So I would not trust it too much. But for instance, it was the case for I think for a transporter protein, let's say in CASP, somewhere I've been analyzing it in the interpretability, and it was indeed kind of doing either this or that and not being sure what to do. For PLDDT, it might show up these regions, but it's not, I would say, the kind of the target use of it. So it's kind of side product where it could be useful.
Very interesting. Thank you. And if I have time for more 2 questions, I would ask them. You have
okay.
Maybe one would be very significant for me. For example, you have some training set, and you know that you have some set of domains or some set of very good known regions of some protein. And how does your AlphaFold performs on the domains it did not seen in a training set, and what can you do to improve prediction on some domain if you know that on this very domain your performance is not very good?
Okay, it's kind of I hear like multiple questions in one a little bit. I mean, start with like you can like starting from the end of the question, like how to tweak something if you're really caring about some particular domain which is not confident in the predictions. I would say sometimes tweaking MSA could help a bit, like resampling MSA or trying to. So like if there are multiple domains in a protein, it could, for instance, be an unlucky case where you don't see enough MSA sequences for this particular domain. So you could try to boost it a little bit, but I don't have like any confirmed measures numbers for this approach. Other than that, I don't see straight away much what you could do for that.
Mm-hmm. And what about domains that you did not see in the training set? How does? Mm-hmm.
Yeah, but that should be fine. So actually, we like it's kind of about like so like we have not seen all those structures from CASP in our training set, and also like in some other research that we've been doing where we were interested to generalize to novel proteins, where we filtered out everything kind of reasonably similar to the training set. We've seen good generalization. So generally assume that's kind of a question about generalization of the method to novel folds, and I do expect it does work reasonably well.
Okay, that's very cool.
Okay.
Thank you.
Thank you. Let's move on. The next question was from Marin, please.
Yes. Hi. I had a question regarding the ligands. So you can't model with the ligand, right? But yet you see side chains being coordinated as if they were coordinating ligands. So I was wondering if there's some sort of bias maybe introduced by the database you use. Like obviously proteins crystallize better if they're more stable. So if they coordinate ligands, they're often stabilized. Is that why you see that? And I mean, somehow it also gives a wrong output because without the ligand, the structure would be different, right? So you can only see one state. Yeah.
Yeah, definitely. I think honestly, there is like still a lot of work to be done. Let's say on ligand binding, because as you're saying, there might be multiple states, and of course, since we are outputting just one prediction or even 5 predictions, we can't really control well which state do we get. So this slide is more like kind of anecdotal example that look like we have not seen the ligand in training and we have not cared about it, but we still got the side chains to coordinate it. I think one maybe related observation is that the final embeddings from the network seem also to be already carrying the information about ligand binding and ligand positioning really. So it might be possible, I think, to kind of fine-tune the network to just explicitly try to extract that. Why it's the case and whether it's like some data bias, hard to say. So we are just,
I don't know if it was in the paper that it said it's worse in predicting NMR structures. I feel like I read that somewhere. So is it biased towards whatever crystallizes?
So we're just sampling from PDB. So of course, like any biases which are in PDB, which of course is coming like from experiments, can play a role. Although I would still assume we do have structures without binding there as well. So we've had some discussions about, for instance, yes, if we are seeing both state of some protein in the data, will we bias to one of them? And it's all a little bit vague, to me, honestly. I would say like we can't really control for this explicitly. So I think it's a cool kind of next challenge to really control it and not like just, look like it worked.
And if I rerun my protein without certain domains, like if I have a pro protein, for example, I cut off one part, would you think that the prediction changes? Because if it's predicting based on the relationship of each residue and then some aren't there, I could get a different prediction? Or? You
could get, Again, it's a little bit hard to control, but you definitely can get it. Like there is a possibility that you will get a different prediction. We had a lot of experiments with this T044 target, the large RNA polymerase, which had like 7 domains or something in the CASP competition. So we tried to fold it by domain, or we tried to search MSA as a whole chain, then cut it and fold, then reassemble and do it like in different order. So we've been getting a little bit different predictions. And in the end, we concluded that for that one, it was best just to fold it end to end as a whole big chain. Exactly for the motivation that you mentioned, that there is more context to use. But if you're trying to tweak something and like not happy with some particular domain, actually answering to the previous question as well, it might be indeed a good idea to just try to cut it and see what happens.
Okay, thanks.
Okay, thank you very much. Let's move to the next question. Yes, Andreas, please.
Thank you. Anna, this is obviously very, very impressive what your team achieved here. I did my PhD on protein folding 25 years ago, and needless to say, it's great to see the progress. I had a question relating to about protein complexes. Now you've been involved also in the AlphaFold multimer sort of implementation, and I was wondering, can you share with us a little bit what some of the maybe obvious and less obvious challenges are to predicting, say, complexes between two proteins? Right, much biology happens by proteins interacting with each other, and if you have a protein complex where biochemically that it's a 1-to-1 or a 2-to-2 complex, what are the challenges of trying to predict an interaction like that? Maybe you can say a little bit on your AlphaFold multimer sort of approach.
Thank you very much. Yeah, and thanks for sharing. And it's like great. To see like people really in this domain for a long time. Since as I mentioned, I've been just coming to this recently. So first, indeed, worth noticing that we've released AlphaFold multimer recently. So there was a bioarchive paper and AlphaFold open source code for this one as well. So AlphaFold multimer attempts to predict complexes as a whole. So in this case, indeed, taking into account all chains, there is nothing about ligands or any other bits there, but multiple chains, homomers and heteromers, can be predicted. So we do think that the current system is already useful for certain biological applications, but at the same time, indeed, as you say, there are important challenges still remaining. For instance, one concrete challenge that we are seeing is that sometimes it's kind of more prone to clashes, to stereochemical clashes, and sometimes we do observe kind of multiple domains clashing together in the center without being properly distanced. We actually currently looking to this a bit more, and we have a bunch of kind of ongoing hypotheses why it might be the case, and we'll probably follow up at some point with the community. But to give a little bit a broader answer, I've realized I actually have another slide on what's coming next. So this AlphaFold multimer release is falling into the first group of extending towards structural interaction map and anything related in terms of interactions with different bits of the biological structures. The second area would be around extending the Lddt database. That one I've already covered. And the last one is probably to deal with what we've been discussing now in Q&A again about multiple conformations, stability, mutants, and things like that.
Okay, let's move on to the last raised hand by Oleg, I guess.
Yes, thank you. I'm the original Oleg. You can say several, but first of all, thank you very much for your presentation. It's really exciting. I have really broad question, but you can just answer as deep as you would like to. So when you show those nice plots of these single proteins, do you understand that it's actually something like low rank MSA representation? So you can basically see which blocks contributed efficiently to a model during backpropagation, or is it just a simulation you made afterwards? It's like talking about explainability of your model.
Okay, so just to confirm, is it this slide that you mean?
Yes.
So those single proteins in the bottom are actually the concrete protein structures that we have predicted with auxiliary structure module heads that we trained particularly for this purpose, for the interpretability purpose. So we have taken our state-of-the-art model. We have decided that all the weights will be frozen in the main network. And then we've added extra structure modules with their own weights. We've allowed them to be trained, again, with backpropagation from the real structures on the PDB examples as usual. And then we applied this inference to a few proteins, for instance, from the CASP14 competition that we were interested to look at. And we've got those single structures, right? So those are predicted by our network, but from intermediate layers from intermediate iterations. Is that answering your question? I'm not sure. And if not, feel free to follow up.
Yeah. So and obviously you have like a ground truth for those proteins. You know exactly how they look like, right?
That I guess I mean now we do, but like we didn't use that for training of this thing, right? So we again trained it on our train set, which is PDB before CASP13, and then we applied it to CASP14 test set just to see how those intermediate representations will look like.
Yeah, I see. That's great, because I think like for most of those like who working actually in the biomedical field, it's like really important to know what they actually see. Yeah. Okay. That's awesome. Yeah. Thank you very much.
Thank you. Yeah, that's interesting. I think it would be really actually interesting to take someone from the biomedical field and like together to look through those kind of videos, because yeah, I've spent quite a while kind of observing how like bits are moving around. For instance, one observation was that something similar to domains would usually be moving together. So like at the early stages, you will decide what are kind of related chunks, and then they will try to move together until they settle. But I guess someone who looks at proteins more could maybe see some more insights. Oh, I see 2 other hands raised.
Okay, yes, but there is also a question in the chat that was asked before, from a question from Mikhail regarding systematic errors, pitfalls of AlphaFold. Can you see it?
Yeah, I can see it now. I can read it. Mm-hmm. Are there known systematic errors, pitfalls of AlphaFold, like protein domains where it systematically produces residue overlaps or something else? That's a good question. So just kind of trying to see if I can kind of generalize our experience in some way and summarize. I guess, well, one I've already mentioned, like on the multimer system, for instance, we are seeing more clashes. We don't see normally clashes on the monomer system, so not there. Any other errors? So we know that like for some certain classes of proteins, AlphaFold is performing a bit less convincing than for others. I guess I won't name those just now, just simply because like kind of I'd like to like refer back to a list, and if you contact me by email, I can bring it back to you, and don't want to rely on my memory. Other than that, I guess we've spent some time on calibrating our confidence heads. So I think in the released models, we do believe that our confidence heads are well calibrated, and there are paper figures kind of showing that confidence of this PLDDT means that, which was not necessarily the case in our earlier models, but that's kind of a pitfall that I think we've addressed. So yeah, that's all that comes to my mind right now.
Okay, thank you very much.
Thank you.
Right. Yeah, thank you. And let us get back to Nazar. He has one more question, I guess.
I have a tiny question. How does it take to retrain the whole model from scratch?
So we have those numbers in the paper as well. So we take about 2 weeks, up to 2 weeks, to train the whole model from scratch on TPUs, on 128 TPU cores. We actually do it in like 2 rounds. We first train and then fine tune, again with details in the supplements, but that's not super interesting, like conceptual at least, just kind of technical detail.
Okay, thank you.